Hi, my name is Dr. Katie Burnett, and I'm going to demonstrate the steps of a bacterial transformation. This tube contains competent E. coli cells. I will label the tube with my initials so I can identify it later. This tube contains the P-Glow plasmid DNA that we transformed into the bacteria. I'm going to adjust my P20 to 5 microliters. Your experiment may contain a different amount of P-Glow. Check your protocol to determine how much plasmid you need to use. After the P-Glow is added to the cells, I will stroke the tube gently to mix and place it on ice for 10 minutes. This allows time for the DNA to stick to the cells while the cold prevents the cells from dividing. I carry the ice bucket with the cells to the heat block set to 42 degrees Celsius and place the tube in the beads and start my timer. This heat allows the plasmid DNA to enter the bacteria cells. After 30 seconds, I'll remove the tube from the beads and place it back on ice. Then I'll return to my bench. SOC is a very rich liquid medium that allows the bacteria to recover from the transformation step and begin dividing. The cells will also start expressing the resistance gene. Check your protocol for the exact amount of SOC to add. The tube is placed in a container along with the tubes from the rest of your class and placed into the shaker. Now you can see why labeling the tube is very important. The shaker is at 37 degrees Celsius, which is the optimum temperature for E. coli. Shaking the tubes keeps the liquid aerated. After shaking for at least 1 hour, the tubes are removed and the cells are ready to be plated on LB medium. There are 5 plates for this experiment. They are labeled with different colored stripes. One black stripe indicates the media is LB with nothing else added. A red stripe indicates ampicillin was added. The sugars added are indicated by the blue stripe is for arabinose, the green stripe is for galactose, and the orange stripe is for lactose. These are the 3 sugars that can activate gene promoters. To spread the cells on the plates, I will add 3 to 4 sterile glass beads to each plate. Next, I will add the cells to one plate. Check your protocol for how many cells to add. To spread the cells, I will shake the plate back and forth so the beads make this sound. If the beads sound like this, you're doing it wrong and the cells are not being spread across the medium. Now I'll finish plating the cells and shake the remaining plates all at once. Notice that the trash pail has a red liner. E. coli is considered a biohazard and is treated as hazardous waste. The red bag will be sterilized before disposal. The beads are removed from the plates. They will be washed, sterilized, and reused in future experiments. Now my plates are ready for the incubator. The plates are placed in an incubator set to 37 degrees Celsius for 10 to 12 hours or overnight to allow colonies to develop. Remember to put your plates upside down. Plates are then removed from the incubator and are ready for analysis. Here is an example of the results. The plate in the top left is covered with a white film of bacteria. Scientists call this a lawn. The small white dots you see on 3 of the plates are colonies of bacteria. Each colony arose from a single bacterium that grew. The number of colonies is what you will be recording. The plates are viewed under ultraviolet light to see which colonies fluoresced green. This phenotype is also recorded.